Okay, let's unpack this. Stop for a second and picture the life teeming around us that we can't see.
Map out the distinct phases they go through in a typical culture and figure exactly what environmental factors are pulling the strings, controlling their speed and survival.
Think of it as getting a backstage pass to the microbial population dynamics show. It might sound a bit niche, maybe,
A little bit.
but these principles, they touch everything from brewing beer to fighting infections.
Mm.
So yeah, let's jump in.
Let's do it.
When we talk about microbial growth, what exactly are we talking about? It's not just one cell getting bigger, is it?
No, that's just the initial part. Fundamentally, microbial growth refers to an increase in the total biomass of the population.
Right, the total stuff.
Exactly.
Mm.
And for most microbes, the main way they increase biomass is through cell division, one cell becoming two, two becoming four, and so on.
Which brings us to that key difference between, like, simple arithmetic increase adding 2 units every hour, say 2, 4, 6, 8,
Linear.
and the kind of growth microbes really do.
Ah, yes, the powerful one. Exponential growth.
Mhm.
You start with 1, then 2, then 4, 8, 16. It's a geometric progression, doubling each time.
This is where the numbers get, well, truly wild, incredibly fast.
Mhm.
The classic way to visualize this is that fable, right? The grain of rice on the chessboard.
Oh, yeah, that one.
Start with 1 grain on the first square, 2 on the second, 4 on the third. By the time you reach the 64th square, the total amount of rice is just astronomical.
It's mind-boggling. It really is. If you actually calculated the number of grains on just the 64th square alone, that's 2 to the power of 63.
Wow.
The total amount of rice involved across the whole board would far exceed anything in a single kingdom, maybe even globally.
It's hard to even picture.
To put it in perspective, stacking that many grains end to end would stretch something like 4.69 times 10 to the 13 kilometers.
It's huge.
The distance to Alpha Centauri, the nearest star system, is around 4 times 10 to the 13 kilometers.
So we're talking interstellar distances from doubling grains on a chessboard.
That's the sheer scale of exponential growth,
Yeah.
just from doubling.
And we even harness this, right? In labs, like with PCR.
Absolutely. The polymerase chain reaction. It uses the same fundamental engine to exponentially amplify tiny amounts of DNA.
Same principle.
Exactly. And the core concept that describes how fast this exponential increase happens is the doubling time, TD. It used to be called generation time.
Okay, TD.
It's simply the amount of time it takes for a microbial population to double in size, or biomass.
And there's a specific equation to describe this process.
Yes, the foundational one is the general growth equation.
Yeah.
X equals xe times e to the power of EAT.
Okay, let's break that down because equations can look a bit scary sometimes.
Sure.
At XD is your starting amount, initial cells or biomass, right?
Yep.
X is the amount you have at a specific time. T is just that math constant base of the natural log.
Mm-hmm.
And then there's that Greek letter, it dot mu. What does it lot represent?
Ah, that's the crucial part. The specific growth rate. It's not just a simple rate, it's a rate relative to the current population size.
Relative. Okay.
Think of it as the fraction of the biomass that's being added per unit of time. Its units are typically inverse hours, like h minus 1.
So kind of like a percentage increase, but it's being constantly applied.
Exactly. Imagine it like compound interest on an investment. If your specific growth rate is, say, 0.5 per hour, means in 1 hour your biomass increases by 50%. But that 50% increase is calculated on the new, larger amount at the start of that hour.
Ah, right. It keeps building on itself.
Precisely. So 1 gram becomes 1.5 grams, then 2.25 grams,
Yeah.
then 3.375 grams. It grows just like money with compound interest. The total increase depends on the rate, the initial amount x, and the duration t.
And we can actually measure this and the doubling time, MTD, from an experiment, like watching them grow.
Absolutely, yeah. By plotting a growth curve, you often see these curves plotted on semi-logarithmic graphs.
A semilog. Why
Yeah.
semilog specifically?
Because plotting the cell concentration or biomass on a logarithmic scale, the y-axis against time on a linear x-axis, it does something really neat.
What's that?
It turns that curve of exponential growth into a beautiful, easy-to-analyze straight line.
Oh, that's brilliant. So much easier than dealing with a curve.
Exactly. A straight line makes calculating the rate so much simpler.
So the slope tells you.
The specific growth rate is simply the slope of that straight line on the semi-log plot.
Nice.
And the doubling time TD is directly related to, it's just the natural logarithm of 2, which is about 0.693 divided by...
Okay, got it. So looking at typical rates then, It seems like prokaryotes, bacteria and such, grow much faster than eukaryotes, like yeasts or molds.
They generally do, yeah. Bacteria can have doubling times well under an hour, sometimes down to 15–20 minutes under really optimal conditions.
Wow, 15 minutes.
Yeasts are typically a few hours, and molds could be even longer.
Is there a simple reason why the smaller cells are often the speed demons?
Yes, it largely comes down to the surface-to-volume ratio.
Ah, that concept again.
Mm-hmm. Smaller cells have a much larger surface area relative to their internal volume compared to bigger cells.
Okay.
This means they can take up nutrients from their environment and get rid of waste products much more efficiently across their membrane. They can basically fuel their growth engine faster.
Makes sense. Faster in, faster out, faster growth. Okay, so we know the math of exponential growth, why smaller cells are often quicker. But that growth rate, it isn't fixed for a given microbe, is it? It depends on the conditions.
Oh, absolutely. What controls how fast they actually grow?
Right.
That's where the environment really comes into play.
Right.
A microbe has a potential maximum specific growth rate, we call it Mumax,
Mumax.
that it can achieve under perfectly ideal conditions.
Yeah.
But in the real world, or even in a lab flask, that rate is usually steered by external factors.
And the source points to several key ones. Temperature, obviously. That seems like a big one.
Huge. Every microbe has a range. A minimum, an optimum, and a maximum temperature for growth. Get too hot or too cold, and mood just plummets.
Makes sense. And then there are other physicochemical factors.
Yeah, things like pH, the acidity,
Mm-hmm.
the amount of oxygen or carbon dioxide dissolved in the liquid, and something called water activity.
Water activity. That's like how much water is actually available for them to use.
Pretty much, yeah. How much unbound water there is. Each microbe has its sweet spot for all these things.
Plus, you have to worry about inhibitors, right? The things that slow them down.
Definitely. These could be substances already in the medium or quite often toxic metabolic byproducts the microbes produce themselves as they grow.
Like ethanol in a yeast fermentation. That eventually stops it.
Exactly. Or lactic acid from certain bacteria.
Mm.
Those build up and can really put the brakes on growth.
But often the biggest factor controlling the rate in a specific situation is the availability of the primary growth-limiting substrate.
Yes,
Yeah.
this is absolutely key. It's the one essential nutrient that is present in the lowest concentration relative to what the cell actually needs for growth at that moment.
So it acts like the bottleneck.
Precisely. It's the one resource that's holding everything else back from supporting that maximum growth rate to max.
And there's an equation for this too, to describe how the growth rate responds to this limiting nutrient.
There is, yeah, and it's foundational. The monod equation.
Okay.
It looks like this. E equals Mayer max times ES divided by the sum of Ks plus S. So Mayer moic is Nair max.
Okay, that formula, it looks kind of familiar somehow.
It should. It's directly analogous to the Michaelis-Menten equation.
From enzyme kinetics. Right?
Exactly. So instead of reaction velocity V and maximum velocity Vmax depending on substrate concentration, you have the specific growth rate and maximum growth rate depending on the limiting substrate concentration, which is IS.
Okay. And just like the Michaelis constant kilometer, there's the monod constant, Vmax. What does that tell us?
So the monod constant, it's the concentration of the limiting substrate at which the specific growth rate is exactly half of the maximum rate.
Half max, okay.
And the key thing is a load case value means the microorganism can still achieve half of its maximum growth rate even when the limiting nutrient is at a very low concentration.
Meaning it's really good at grabbing it even when there's not much around.
Precisely. It indicates the microbe has a high affinity for that substrate. It's very efficient at scavenging it from a dilute environment.
So the monod equation essentially shows that as the limiting nutrient concentration crust goes up, the growth rate also goes up, but it eventually levels off. It saturates at Nomax once the substrate isn't limiting anymore.
Exactly right. It's a really powerful model for understanding how nutrient levels constrain microbial speed.
Now, imagine we take some microbes, put them in a closed container with a fixed amount of food, like a flask in the lab, a batch culture.
Okay, yeah.
They don't just follow that monod equation and grow exponentially forever, do they? They go through a whole life cycle, sort of.
They absolutely do.
Yeah.
What you see is a standard microbial growth curve. People have been documenting this for over a century, really. Though foundational work by people like Manon back in the 40s helped clarify the underlying principles, especially balanced growth during that exponential phase. Batch culture experiments are fundamental tools. Absolutely. But interpreting the data correctly requires understanding these distinct phases they go through.
So the first phase, right after you introduce the microbes to the fresh medium, that's the lag phase. Often, nothing much seems to happen for a bit. Why the pause?
Yeah, the lag phase. Well, there's several reasons. The cells need to adapt,
Yeah,
adapt to the new conditions.
makes sense.
They might need to synthesize new enzymes to actually use the specific nutrients you've given them. If they're coming from a different environment, maybe they need to repair some damage or detoxify inhibitory stuff in the new medium.
Okay.
And there's also evidence that even for aerobes, the dissolved CO2 levels might need to build up slightly in the liquid.
Really? CO2? Even for aerobes?
Yeah, CO2 is actually required for certain central metabolic pathways, these anafluorotic reactions. So they might need a bit to get going properly.
Huh. And I found it fascinating that the source mentions bacterial communication playing a role here too. Like they might be sensing their environment or even their own numbers using signaling molecules.
Yes, that's a really active area of research. Quorum sensing and related phenomena. For some species, there's evidence they might need to secrete specific signaling molecules, sometimes called pheromones,
Pheromones.
or reach a certain threshold concentration of these signals before they really kick off rapid growth.
So they're waiting for a signal, almost.
In some cases, yeah. The example of Micrococcus luteus needing a secreted protein, or the finding that adding cell-free liquid from an actively growing culture can shorten the lag phase, especially in minimal media that strongly suggests this kind of communication is happening.
And the length of this lag phase, it's not random, right? It can vary quite a bit.
Oh, certainly. The microbial species itself matters some, like certain mycobacterium, are known for having notoriously long lag phases. And crucially, the growth phase of the cells you use to start the new culture. The inoculum that's hugely important.
How so?
Cells taken from the actively dividing exponential phase will typically give you the shortest lag. They're already primed to grow.
Okay, that makes sense. And surprisingly, the size of the inoculum matters too. More cells means shorter lag.
Up to a point, yes. Generally, a larger inoculum tends to lead to a shorter lag phase.
Why would that be?
Well, it might be related to them collectively reaching a sufficient concentration of those internal metabolites or external signaling molecules faster.
Ah, like hitting that quorum faster.
Exactly. Or maybe just building up necessary CO2 levels more quickly. So if you're trying to start a culture quickly in the lab, using a fresh inoculum from late exponential phase and, an appropriate starting density can make a big difference.
Good practical tip. Other factors mentioned are using a rich medium.
Yep. Shorter lag usually.
Being at the optimal temperature and having enough dissolved CO2.
All help get things going faster.
Okay, so after the lag, you often see a short acceleration phase.
Yeah, just a transition where the cells are fully switching on their growth machinery.
Yeah.
And the specific growth rate, LEC, starts increasing towards its maximum.
And then the main event, the exponential growth phase. This is where you see that constant, fastest rate of doubling.
This is the period where, theoretically, the specific growth rate is constant and equal to mu max. Because all essential nutrients are present in excess of what's limiting, according to that monad model we talked about. Nothing's holding them back, nutritionally speaking.
Right, full speed ahead. It's interesting, though, that even during this fast growth, some microbes will still excrete things like acetate from E. coli, even with plenty of glucose around.
Yeah, overflow metabolism. It happens.
And they might accumulate storage compounds, too, like saving for a rainy day.
They do. Microbes are complex. Sometimes they grow so fast they can't metabolize everything perfectly efficiently, or maybe they're anticipating future lean times.
So they store stuff.
Yeah, accumulating intracellular storage polymers like glycogen or PHB, polyhydroxybutyrate, or polyphosphate. It happens, especially when they have lots of carbon source like glucose, but maybe you're limited slightly elsewhere. It's like putting reserves in the bank.
Okay. But eventually, that exponential party has to end. As resources dwindle or waste products build up, the growth rate starts to slow down in the deceleration phase.
Yep, that's the next stage. This phase begins when the factors supporting Mmax are no longer fully satisfied.
What usually triggers it?
The most common triggers are either the concentration of the primary growth-limiting substrate dropping low enough, typically below about 100 times its case value,
Okay.
or the accumulation of those toxic metabolic products we mentioned, like ethanol from yeast or organic acids from bacteria, reaching inhibitory levels.
Right, so either running out of food or poisoning themselves.
Basically, yeah. Though if substrate limitation is the only factor, this deceleration phase can sometimes be very brief, almost unnoticeable.
And as they enter this slowdown and prepare for tougher times ahead, the cells undergo some pretty remarkable structural and physiological changes, right? They really transform.
Dramatic ones. They essentially batten down the hatches for survival. There's a massive decrease in overall gene expression. Protein synthesis slows way down.
Makes sense, conserving energy.
And they swap out their lower affinity nutrient transport systems for high affinity ones.
To scavenge better.
Exactly, to grab the very last bits of substrate available.
And they can really bulk up on those reserves you mentioned earlier, like lipids or capsules.
Oh yeah, especially when one nutrient, like nitrogen, becomes limiting, but there's still plenty of carbon source around. Yeasts can accumulate huge amounts of lipids.
Wow.
Some bacteria, like Klebsiella pneumonia, can produce these massive polysaccharide capsules. The capsule can actually make up like 60% of the cell's dry mass.
60%. Just capsule.
Yeah. So this adds to the total biomass, even if cell division itself is slowing right down.
And their cell walls change too, getting tougher.
Absolutely. They often thicken and become more resistant to autolysis, that's self-digestion.
Okay.
Their actual composition can change too. For example, Bacillus species swap out wall components called teichoic acids for something called tituronic acids when phosphate gets scarce.
Adapting the wall itself.
And they also increase the cross-linking in their peptidoglycan layer, which makes the wall stronger, more rigid.
Plus, membranes become less permeable, cytoplasm gets denser, and they often become smaller and stickier, more hydrophobic.
Yeah, that increased hydrophobicity might help them adhere to surfaces in their natural environment, which could be beneficial for survival.
All these are adaptations for just surviving, basically.
Exactly. And physiologically, they develop increased resistance to various stresses, heat, oxidation, osmotic changes, often by producing specific stress response proteins. And there's this amazing finding mentioned. When energy gets severely depleted, some cells can even protect their DNA through a process of reversible biocrystallization.
Reversible biocrystallization of DNA. That sounds like something straight out of science fiction.
It does, doesn't it? It really underscores how incredibly hardy these stationary phase cells can become.
So they're much tougher than the ones actively growing.
Generally, yes. Cells from the stationary phase are far more resistant to environmental damage. They survive preservation methods like freezing or drying much, much better than those fragile, rapidly growing exponential phase cells.
That's a really critical practical point if you're working with microbes in a lab or industry.
Absolutely vital.
So after slowing down and transforming, they enter the stationary growth phase. The term implies everything just stops, but is that actually accurate?
Well, it's defined by a constant viable cell concentration.
Meaning
Meaning the number of cells capable of dividing and forming a colony on an agar plate isn't increasing significantly anymore. But even then, different things can be happening under the surface.
like what?
Sometimes the total biomass is constant, but the number of cells might initially increase just a little bit. Because cells that were in the middle of dividing during the exponential phase might finish the process, resulting in slightly smaller cells overall. So number goes up slightly, but total mass stays put.
Or is it possible that cells are actually dying, but others are eating them, keeping the numbers steady?
Yes, exactly. That's a phenomenon called cryptic growth.
Cryptic growth.
The total cell count might appear stable, or the viable count might plateau while the total count stays constant, because some cells are lysing, breaking open, and the nutrients they release are being used by other survivor cells to continue growing, basically balancing out the death rate.
That makes it tricky to know exactly when stationary phase really begins, especially if you're just measuring, say, optical density, which counts total cells, dead or alive.
It is quite tricky, yeah. Total count methods can be misleading there.
And sometimes they might switch gears and start using those byproducts they excreted earlier, like acetate, as a food source once the main course is gone.
Right. They can be quite resourceful. And then there's that fascinating case mentioned in the source. Some starved cells, like Micrococcus luteus.
Yeah.
When you transfer them to fresh medium, they only divide a few times and then just stop growing, even though there are plenty of nutrients.
Why would they do that?
It was discovered they actually secrete a killer factor that inhibits further growth of their own kind.
Whoa. Another potential form of growth-regulating signaling or population control.
Possibly, yeah. The strategies these microbes employ are just incredible.
But eventually, even in stationary phase, the viable cells do start to decline. You get the accelerated or exponential death phase. They lose their ability to be culturable. You can't plate them and get colonies anymore.
Right. And there are a few hypotheses floating around for why this happens.
Okay, what are they?
One idea is a form of programmed genetic death, like cellular suicide. Another proposes they enter a viable but non-culturable VBNC state.
VNC, meaning they're still alive?
Yeah, still alive, still metabolically active, but for some reason, they just won't grow on the standard laboratory media we use to count them. It might be a kind of deep dormancy, a survival strategy in really harsh conditions.
Okay. And the third hypothesis sounds, well, surprisingly relatable, something about aging.
Yes, it's quite thought-provoking, this one. This hypothesis suggests that starvation, over a long time, leads to damaging oxidative stress inside the cell.
Oxidative stress, like rust, but for cells.
Kinda, yeah. Damage from reactive oxygen species. In stationary phase, when cellular processes slow down and maybe repair mechanisms aren't working at full tilt, you can get an accumulation of damaged proteins. This can be followed by irreversible damage, particularly a process called carbonylation of proteins.
Carbonated proteins, that sounds bad.
They're trouble, yeah. These damaged proteins can aggregate, clump together inside the cell, actually similar to the protein aggregates observed in neurodegenerative diseases in humans, like Parkinson's or Alzheimer's.
Wow, really? In bacteria?
Yeah. This accumulation of irreversible damage to key cellular components, ribosomes, proteins in the membrane, the periplasm eventually leads to the collapse of cell function and ultimately death.
So this oxidative damage idea suggests a fundamental mechanism of cellular decline and death in microbes could actually share surprising parallels with aging processes we see in complex organisms, like ourselves.
It's a really striking connection, isn't it?
It really is.
And the specific patterns of death can vary, too. Sometimes the viable count drops while the total count stays stable, maybe due to that VBNC state, or just slow lysis. Other times, both drop quickly because of widespread autolysis, cells just falling apart.
And things like temperature and toxic compounds would speed this up, presumably.
Oh, absolutely. They influence the death rate significantly.
It just highlights again how dynamic a batch culture is, constantly changing, and how incredibly adaptable microbes are to these shifts.
And it reinforces the point that it's just absolutely critical for anyone working with microbes. You must know the growth phase or the specific environmental conditions that are limiting your cells when you do an experiment or take a measurement.
Why is that so critical?
Because using cells from exponential phase compared to stationary phase will give you completely different results. Their physiology, their stress resistance, their protein content, their gene expression, it's all dramatically different.
So results wouldn't be comparable or even valid if you didn't know the state of the cells.
Exactly. It drastically impacts data validity and comparability, which is why, for research that needs really consistent cell physiology, continuous cultures, like a chemostat, are often preferred.
Ah, the chemostat.
Right.
Precisely. In a chemostat, you're continuously adding fresh medium and removing culture broth so you can keep the culture in a steady state. Often it's like a continuous exponential growth, though not necessarily at the maximum rate, Micmax. Okay, now speaking of growth that isn't always that clean exponential curve, we mentioned that batch culture has all these phases. Is all microbial growth fundamentally exponential when conditions allow? Or can it be different?
That's a great point. While exponential growth, where the specific growth rate is constant and the log plot is that nice straight line, is the ideal under excess conditions, growth can also be linear.
Linear growth. So the specific growth rate isn't constant anymore.
Right. In linear growth, Bayes actually decreases over time, but the amount of biomass increases at a constant rate. So you get a straight line on a standard plot of concentration versus time, not a semi-log plot.
Okay, when does that happen? When do you get linear growth?
Linear growth occurs when the primary growth-limiting nutrient isn't just low, it's being supplied to the culture at a constant rate.
Ah, okay, that makes sense. If the input rate of the bottleneck resource is fixed, then the output rate, the biomass increase, might also become fixed, regardless of how much biomass is already there.
Right, exactly. Good examples of systems where this happens are fed batch cultures, where you deliberately pump in a limiting substrate solution at a constant rate.
Okay.
Or, as we just mentioned, chemostat cultures. Now wait, this is slightly subtle. While the cells inside the chemostat are often in a physiologically exponential state, meaning they're doubling at a constant rate set by the dilution.
Right.
The total number of cells in the vessel itself only increases linearly over time if you consider the whole system, including the outflow, because cells are being constantly washed out at that fixed rate determined by the fresh medium flow. If you stop the outflow, it becomes batch again.
Okay, gotcha. So the accumulation in the vessel under continuous flow can be linear. And you also mentioned aerobic cultures under oxygen limitation.
Yes, in a batch system like a shake flask. We'll get more into oxygen soon, but because the rate oxygen transfers from the air into the liquid, the OTR,
Yeah.
is often constant once it becomes limiting.
Uh-huh.
That constant supply rate of the limiting nutrient, oxygen, can lead to linear biomass increase for obligate aerobes.
Okay. The source also mentioned a less intuitive cause. Conservative or luxury uptake of trace nutrients. How would that cause linear growth? That sounds complicated.
It's a bit more niche, yeah. The idea here is that some microbes can take up certain trace elements or vitamins way in excess of their immediate needs when they're available. That's the luxury uptake.
Okay, hoarding nutrients.
Kind of. Then they might transfer these crucial but tiny amounts of stored nutrient to the daughter cells at a constant rate per division, or maybe per unit of time.
Right.
So if the overall growth rate is ultimately limited by the internal concentration or supply rate of this slowly transferring nutrient, and that internal supply rate stays constant due to this uptake and transfer mechanism, then the growth rate per cell becomes constant. And if the addition of the limiting factor to the growing part of the system is constant through this internal doling out, it can result in a linear increase in total biomass for the population.
Wow, that's fascinating how internal cell processes, like how they share stored goodies, can influence the overall population growth pattern.
Absolutely. It highlights the layers of complexity.
Okay, let's focus in on one specific limiting nutrient that you mentioned presents unique challenges. Oxygen.
Ah, yes. Oxygen. Absolutely essential for many microbes, for aerobic respiration, which gives a huge energy yield. But it's also really tricky to manage in cultures.
And microbes have very different relationships with it, don't they? There are different categories.
That's right. You have your obligate aerobes that absolutely need it to live, then obligate anaerobes that are poisoned by it.
Right.
You have aerotolerant ones that can survive oxygen but don't use it for energy. Facultative anaerobes, like E. coli or the yeast Saccharomyces cerevisiae, are flexible. They prefer oxygen but can switch to fermentation if it's absent.
Like brewer's yeast.
Exactly.
Yeah.
And then microaerophiles, which need oxygen but only at low levels, high levels can be toxic to them.
And oxygen's effect on growth, it also follows a monad-type relationship.
It does, yeah. The growth rate depends on the dissolved oxygen concentration, let's call it C, and a specific constant, Ko, which reflects the organism's affinity for oxygen. Similar idea to case for substrates.
And there's a point where oxygen becomes truly limiting for aerobes, right? A critical level.
Yes, the critical dissolved oxygen concentration, often called secret. Below this level, the dissolved oxygen concentration is simply too low to support the maximum possible respiration rate.
So oxygen supply becomes the bottleneck.
It does. And secret values vary between organisms. They can be quite low for some, like maybe 0.12 to 0.15 milligrams per liter for Saccharomyces cerevisiae.
Okay.
Or a bit higher, around 0.26 milligel for E. coli. Still very low concentrations, though.
And the main challenge with oxygen in liquid cultures is its very low solubility, right? It doesn't dissolve well.
Exactly. That's the crux of the problem. It only dissolves at around maybe 7 milligrams per liter in typical culture media at room temp and pressure.
That's practically nothing compared to how much sugar or nitrogen they might consume.
Precisely. Which means for any actively growing aerobic culture, you can't just rely on the oxygen initially dissolved. You have to continuously supply it from the air.
Which you do by shaking a flask or bubbling air, sparging, into a fermenter.
Right. And the rate at which oxygen can actually move from the gas phase, the air bubbles or headspace, into the liquid phase where the cells are. That's called the oxygen transfer rate, or OTR.
OTR. And often that OTR is the actual rate-limiting step for growth in aerobic cultures, more so than the cell's ability to use oxygen.
Very frequently, yes. Especially in simpler systems like shake flasks, or even in large fermenters if the mixing or aeration isn't good enough. If you've ever stopped shaking a flask of rapidly growing aerobic bacteria, you'll notice the dissolved oxygen level, if you measure it, drops to basically zero incredibly fast. Sometimes in just seconds.
Wow. They just suck it all up.
Instantly.
Yeah.
So walk us through a typical shake flask scenario. You start with an obligate aerobe. You start with some dissolved oxygen in the fresh medium. The cells start growing, consuming it. As the cell concentration increases exponentially, The rate at which the whole culture consumes oxygen, that's the volumetric uptake rate, QO2, also increases exponentially.
Makes sense. More cells eating oxygen faster.
Right. Meanwhile, the oxygen transfer rate, OTR, from the air into the liquid, driven by the shaking and diffusion across the surface. That rate is relatively constant, assuming the shaking speed and flask setup don't change, especially once the dissolved oxygen is near zero because the driving force for transfer is maximized then.
Okay, so you have QO2 going up exponentially and OTR staying roughly constant.
Exactly. So eventually QO2, which is rising fast, will exceed the constant OTR. The culture needs more oxygen than the flask can physically supply per minute.
And what happens then?
The dissolved oxygen concentration inevitably drops below secret. Growth is now limited not by the microbe's potential, but by the constant rate at which oxygen is physically entering the liquid.
The constant rate limiting nutrient supply again.
Precisely. Since the rate of the limiting nutrient supply, OTR, is constant, the rate of biomass increase can no longer be exponential. For an obligate aerobe in this situation, the growth becomes arithmetic or linear.
Got it. So oxygen limitation in a shake flask is a classic cause of that linear growth we talked about earlier.
It is for obligate aerobes. Now, slight difference. If you have a facultative anaerobe under oxygen limitation,
Like yeast or E. coli.
yeah, they have another option. They can often switch their metabolism from aerobic respiration to fermentation.
Right.
This means they might actually continue to grow exponentially or pseudo-exponentially, even without enough oxygen. Though likely with a different biomass yield per unit of carbon source, because fermentation is much less energy efficient than respiration.
Okay, so their growth pattern might look different under oxygen limitation compared to an obligate aerobe.
It often does.
Yeah.
And just a practical tip from the source for getting more oxygen into a shake flask.
Yeah.
Keep the liquid volume relatively low compared to the total flask volume, maybe only 10–15%,
Why low?
to maximize the surface area for gas exchange relative to the liquid volume. And use baffled flasks if you can.
Baffled? Those ridges inside.
Yeah, those ridges create turbulence, splash the liquid around more, and significantly improve the oxygen transfer rate compared to standard smooth flasks.
Good tips. Okay, wow. We've covered a lot of ground. The math, the life cycle, the controllers, special cases like oxygen. Why should someone listening care about all these specifics? How does it connect back?
Oh, it connects everywhere.
Oh.
Understanding these fundamental principles of microbial growth kinetics is absolutely critical in countless real-world applications. It's not just academic.
Think about fermentation, right? Whether you're making yogurt, cheese, beer, wine at home or industrially.
Yeah.
Or producing valuable compounds like antibiotics or enzymes or biofuels in industrial biotechnology. Controlling microbial growth rates and phases is completely essential for efficiency and getting the product you want.
Absolutely. It's also vital in wastewater treatment. Those plants rely on carefully managed microbial communities to break down pollutants.
Yeah.
You need to know how fast they grow and what they need.
And understanding how quickly pathogens multiply using these same principles is obviously key in infection dynamics in medicine.
Critical. And it's fundamental to food spoilage prevention and food safety. Knowing the conditions that promote or inhibit the growth of undesirable microbes is basically how we keep food safe to eat.
Yeah, controlling temperature, water activity, pH, it all comes back to controlling mood. And as we discussed quite a bit, the physiological state of the microbes you're working with, whether they're in rapid exponential growth or scavenging in stationary phase or even entering the death phase, that drastically impacts their properties and their behavior.
Hugely important. That's massive for reproducibility in research or for optimizing an industrial process where you need consistent performance from your microbial workers.
So knowing these principles really empowers you, the listener, to make sense of complex biological systems and maybe tackle major challenges like developing new antibiotics or combating antibiotic resistance, which is often linked to the growth state of bacteria.
Very true. Dormant or slow-growing cells can be much harder to kill.
Or designing more efficient and sustainable bioprocesses for making chemicals or materials.
Exactly. It's not just theoretical. It has really tangible impacts on health, industry, and the environment around us.
Well, it's been a really deep dive today into this unseen world of microbial population dynamics.
It really has. We've peeled back quite a few layers, haven't we?
Yeah. From the exponential power of doubling and the math that describes it, through the dramatic life story of microbial batch culture with all its distinct phases and clever adaptive strategies, to the critical environmental factors that control their growth speed, including those special cases like linear growth and the unique challenge of getting enough oxygen.
It truly reveals the Well, the sophistication, the complexity of how microbes sense and respond to their ever-changing conditions, always trying to optimize their survival and growth. They're amazing little machines.
They really are. And for a final thought to leave you with, drawing from our source material today, consider that really striking hypothesis we discussed.
Yeah.
That microbial death, at least in part, might be driven by starvation-induced oxidative damage and the aggregation of damaged proteins, much like processes implicated in aging and certain diseases in complex organisms like us.
That parallel is quite stunning.
It is. So what could this shared vulnerability, these potential parallels in fundamental cellular breakdown processes across incredibly different forms of life, what might that tell us about the universal mechanics of biological decline, maybe, and resilience itself? It's a pretty profound question, starting right down there with the smallest forms of life.